Okay, so welcome back friends. Welcome back in another video tutorial from microbiology with Shagnik. So this is going to be the third video lecture of application in recombinant DNA technology, where we are going to see the blessings of recombinant DNA technology, another blessing of the recombinant DNA technology in the medical field. So in the second video lecture we are discussing about various important role of recombinant DNA technology in medical field, where we have discussed about how we can prepare insulin at a very low cost with the help of recombinant DNA technology. So today we are going to discuss a concept which is based on the same principle, that we have to apply same minimal cost so that we can generate the drug which will of low price, right? So similar thing will be applied in case of the production of the human growth hormone. So before going to discuss about how it will be produced with the help of recombinant DNA technology, it is very important to discuss about what are the importance of human growth hormone and what is it. So human growth hormone, as its name suggests, it is responsible for the growth of the human, right? So from where it is synthesizing? So it is synthesizing from a part of the brain known as pituitary gland. Okay, from pituitary gland, after secretion, it causes growth of the muscle, it causes growth of the bone, and also the lengthening of height. All things are regulated by human growth hormone. And when from pituitary gland the human growth hormone are secreted at a large amount, then it facilitates the condition called acromegaly or gigantism. And when human growth hormone is secreted from the pituitary gland at very low amount, then a condition called dwarfism occurs. Okay, so in children whose pituitary gland do not secreting or secreting human growth hormone at a very low amount, they are often treated with a treatment in which we are simply breaking the skull of the diseased human, and then we will isolating pituitary gland, and from pituitary gland we are isolating the human growth hormone, and then we will purify them and inject into those children. Especially children of age 1 to 8 are treated with this kind of treatment from age 1 up to age 8. The children should have to take human growth hormone which is of a diseased human. Okay, so that was the previous treatments that was popular when our recombinant DNA technology is not developed as much as it today. So the main two problem of this treatment is treatments is obviously very costly, because I hope you can understand that here we are breaking a skull of diseased human, and from those diseased human we are isolating and purifying the human growth hormone and then injecting into a child's body, and child have to treated with that human growth hormone from the age 1 to age 8, right? So it will be obviously very costly procedure. Not only that, we don't know how this human growth hormone will work on the child's body, because the age of the diseased animal can be anything. It can be a old person, it can be a younger person, or anything, right? So the human growth hormone of that person can show any kind of negative effect on the body of a children whose age is only 1, because Don't forget that in age on, our immune system is not too much developed to fight against something that can be present in a diseased animals, or sorry, this is not diseased animal, diseased humans. So that was a major cause. That means it facilitates the form development of some kind of disease that can be very dangerous. Okay, so now what will be the way out? Because human growth hormone is very important, otherwise the development of the bone or development of the muscle will be halted. And if we are going to treat human growth hormone, or going to inject the human growth hormone of a diseased human, then there can be a chance of the transmission of disease, right? So we have to think about something that will reduce the chance of the disease or totally nullify the chance of the disease. Not only that, we have to also think about something that will reduces the price of the drug also. Okay, so under such circumstances, recombinant DNA technology again help us. So let's see how recombinant DNA technology protecting us from that problem. Okay, so here the procedure all starts after the development of reverse transcription technology. Okay, so with the help of reverse transcriptional technology, we have first developed the cloned human growth hormone cDNA. Now I think you may think about that in case of insulin we do not have to derive the cDNA, but in case of human growth hormone we have developed the cDNA. So what is the reason behind that? So here the idea is, in case of insulin there was no presence of any kind of introns, so we can consider the normal DNA and to produce the insulin. But in case of our human growth hormone, there is a presence of intron, that is our non-coding genes in the DNA which is coding for human growth hormone. That is why we don't consider only the normal DNA. We have to isolate the cDNA molecule, then from cDNA we can proceed. Now here I want to mention you that how cDNA can be produced. So cDNA can be produced from the mRNA, which will code or which will produce the human growth hormone after translation. So our idea will be, we will treat the mRNA with reverse transcriptase enzyme. Reverse transcriptase is an enzyme who have the ability to synthesize DNA by using RNA as a template. Okay, so as a result it will facilitate the formation of cDNA. cDNA means complementary DNA. Okay, so that's how we are producing human growth hormone cDNA. Okay, now I want to mention you another important thing here, that here it is a secretory protein. Okay, that is why it consists of a mammalian signal sequence. Okay, and it also contains a eco-R1 cut site after near about 1 to 24 amino acid. And remember one thing, that human growth hormone is a hormone of 191 amino acid. Okay, and there is the eco-R1 cut site after 24 amino acid, and there is also a Henry cut site, although which is not a part of human growth hormone. There is a stop codon, you can see it, and a fast amino acid of mature human growth hormone. So the idea is, first of all, scientists thought to remove the signal sequence, because we don't need so much signal sequence. Our main thing is that deep green portion, we require this. So why we need signal sequence, right? So scientists thought to clip this with the help of eco-carbon cut site, as we have here eco-carbon cut site, right? So that's how they remove the first 1 to 24 amino acid and take only the amino acid which is from 24 to 191. Okay, now they try to express them in E. coli cell, and they then realized that without 1 to 24 amino acid we cannot create a properly folded protein. Okay, and I have told you in our very first video lectures of recombinant DNA technology that during the expression of the protein, folding is necessary, and the first to 24 amino acid of the human growth hormone responsible for that folding, which is not be providable by the rest of the 24 to 191 amino acid. So under such circumstances, scientists decide to replace those 1 to 24 amino acid with a synthetic oligonucleotide that will fill up the gap of 1 to 24 amino acid. And another thing I want to mention here, that here they have kept a hen 3 cut site intentionally. Why? We are going to see this within some time. Okay, so after the generation of complete 191 amino acid containing human growth hormone, they have developed the vector, and they have treated the vector with hen3. Also they have, so here we don't need to treat them because there is a ready-made staggered in present, right? So we clip them. Here we are only clipping the vector, then ligate them with our gene of interest, right, in a expression vector. So here the promoter again will be a lag promoter because we are using a E. coli system. So if lag promoter we are using, then in the plate there will be presence of IPTG or lactose. Okay, along with other important things such as a appropriate antibiotic resistant gene and growth hormone coating gene should be there. Okay, then we will transform them in E. coli, so for the accommodation of the growth hormone inside the bacterial cell. Okay, so after that we have to purify them, and we will end up with a protein that contain methionine at its N terminal. Okay, so this kind of human growth hormone is known as methionine containing human growth hormone. So what we have done, we have first generated a cloned human growth hormone cDNA. So this cloned human growth hormone cDNA contains a eco-carbon cut site. Okay, so and a signal peptide which is the indication that human growth hormone is a secretory protein. So the first thing is we have to remove the signal peptide. That is why a eco-carbon cleavage is applied to remove the first 1 to 24 amino acid. Okay, then by using just only 24 to 191 amino acid, scientists find out that without 1 to 24 amino acid, properly folded protein formation cannot be possible. So they decided to replace the signal peptide with a ready-made oligonucleotide that will not only fulfill the gap of 1 to 24 amino acid but also providing us a extra cut site for HIN3. Okay, so this HIN3 will later help us during the ligation step. Okay, and as a result we will have our expression vector containing gene of interest. Okay, that will ultimately translate to give the protein. Okay, and this protein, as it contains initiator methionine at its N terminus, we can say them as methionine containing human growth hormone. Okay, now as we are growing or producing human growth hormone inside a E. coli, the purification step can be difficult. because remember, you will not get only human growth hormone after lysis of the cell, but you will also get a mixture of protein sample which will contain all the kind of E. coli protein along with your recombinant protein. Okay, now the idea is, if you have you are using this method that I have discussed now, then the purification step will be very difficult, because the purification of the recombinant protein from those mixture of the protein will not be a joke, right? So as a result, this laborious work during purification will reflect or will affect the price of the drug. As a result, price will be high, and I have told you that during the large scale in medicine production in industrial level, the cost factor is very important, right? If cost factor is very high, then we cannot simply use that procedure, or we can modify them to make it easier, so that the price will be affordable by the normal peoples, right? So now what to do? So I hope you have not forgotten that I have mentioned you that human growth hormone is a secretory hormone. Okay, more specifically saying, is a secretory protein, and that is why it is having a signal peptide. Okay, at its beginning, right? Although the signal peptide is mammalian, so scientists thought that start thinking about that if we can replace those 1 to 24 amino acid with a sequence that will code for a signal peptide which is for E. coli, then we can cause the secretion of the human growth hormone in the periplasmic space. Okay, because don't forget that E. coli is a gram negative bacteria, and in gram negative bacteria there is a presence of periplasmic space, right? So if we are incorporating a signal sequence which is not for mammalian, in this case, which will be here for bacterial, so the E. coli cell will consider the human growth hormone as a secreted protein, and as a result it will secrete it in the periplasmic space. Okay, and now by disrupting the periplasmic space, purification of the protein become easier, because obviously the protein crowd that is in cytoplasm and the protein crowd that is in our periplasmic space, there will be a very huge difference, right? Obviously the protein count in periplasmic space will be much lower than in case of cytoplasm, right? And as a result, the purification process will become easier, and as the purification process become easier, it will obviously cause the reduction of the price, and as a result it can be affordable by the peoples. Okay, and that is how recombinant DNA technology is helping us to produce human growth hormone in a most affordable way. Okay, so now let's see this picture. Here we have bacteria signal sequence extra, and this is our promoter one, that orange one. Then we are transforming them in E. coli. Okay, and now the signal peptide that will be produced by the vector will be cleaved by the periplasmic proteases. Okay, I have seen here the periplasmic proteases will cleave off the signal peptide as it has done in this case. Okay, and as a result, the human growth hormone that we will get will be a methionine less human growth hormone. Okay, because here the initiator methionine at before or at the beginning of the bacterial signal sequence, right? So when it will undergo transcription to produce the mRNA and then translation, and then after inside the cell, due to the presence of periplasmic protease, the bacterial signal sequence will be cleaved, right? And as a result, our initiated methionine also be cleaved, and as a result, the human growth hormone that we have now is a methionine less human growth hormone. Okay, so that is the concept of human growth hormone production with the help of recombinant DNA technology. So the process is exactly same and very simple. So first thing is that why we need human growth hormone. Human growth hormone is secreted from the pituitary gland of the human, and that play a important role in the growth and development of the muscle, bone and length, that is the height, right? So if in some children or in some individual human growth hormone is not producing in accurate amount, so it will facilitate the cause of or the chance of dwarfism. And if human growth hormone is at very large amount, then it will facilitate the cause of what? Cause of gigantism, right? So gigantism is not our concern, but if the pituitary gland is secreting very small amount of human growth hormone or not secreting, then it will be very difficult, because the growth of the muscle, bone and also the height is hampered. Okay, under such circumstances, I want to mention about a very famous football player, that is Lionel Messi, who was suffered with a same kind of problem in his left leg, that is the growth hormone deficiency. Okay, and Lionel Messi have to be treated with this kind of treatment that I have mentioned here, and that was very costly, and that is why Lionel Messi's father have to pray help from Barcelona, right? So that was the idea. Okay, so if at that time recombinant DNA technology is developed in such a way that it is today, then it will be very easy for fathers like the father of Lionel Messi to treat their child's problem, right? So how recombinant DNA technology are saving us day by day. So the idea is very simple. We are preparing the cloned human growth hormone, that is cloned human growth hormone cDNA production first, which consists of a eco-argon cut site at the 24th amino acid. With the help of this cut site, first of all we are removing the signal sequence. Okay, the first try is obviously the expression of 24 to 191 amino acid sequence. Then scientists find out that the protein we are getting by coding the 24 to 191 amino acid is not properly folded. Okay, so they understand that 1 to 24 amino acid is equally important, so they decided to replace it with a oligonucleotide that will not only provide the 1 to 24 amino acid that we have replaced, but also provide a Hindi 3 cut site. Okay, this Hindi 3 cut site will play a important role later when we will ligate our gene of interest with the vector. We will going to show this. Okay, so that is how we are ligating it, then we are transferring it in a E. coli, then it will produce the protein. Now there is a problem that the purification becomes very difficult, because there is a very too much crowd of E. coli protein, and from that you have to isolate your recombinant protein. That is not a joke. And this laborious purification process reflects its effect in the cost. And I have told you that the medicine production should be at less cost so that the price of the drug will be lower. And as a result, we have decided to replace the 1 to 24 amino acid with a oligonucleotide that will be a sequence that will facilitate the production of a bacterial signal peptide. Okay, so this bacterial signal peptide will help E. coli to recognize our recombinant protein as a secretory protein, and as a result it will secrete it in its periplasmic phase, and purification and isolation of recombinant DNA from the periplasmic phase is much easier than the purification in cytoplasm, because remember, the crowd of the protein in cytoplasm is much higher than the crowd of the protein in periplasmic place. Okay, so in the second case, as the bacterial signal peptide is cleaved off by the periplasmic proteases, so here the initiated methionine is lost. That is why the human growth hormone we are having is a methionine less human growth hormone. Okay, but in the first cases we are getting a methionine containing human growth hormone, although the function of human growth hormone in both the cases are exactly the same. Okay, so that was the concept of human growth hormone production with the help of recombinant DNA technology. So I hope this video will be helpful to you to understand the concept of production of human growth hormone. So thank you for listening to this class. We will again come back with another topic of application of recombinant DNA technology, that is the BT crop. Okay, so thank you, take care.